addresses.
We are definitely starting with the Fraser, and it's a huge amount of work, but you know, if we develop something that can be used in other watersheds, absolutely.
Okay, I'm going to have to cut us off there. Sorry, Finn, and I'm going to pass the baton over to Christina Charette. Christina is a colleague at the River Institute. She's a research scientist. She's currently doing her PhD and she's chairing our next session. Sorry for the loss of a couple of minutes, Christina, but I'll give it to you.
Thank you, Lee. So welcome everyone to the Living Waters session of the 2020 River Symposium. Like we mentioned, my name is Christina Charette. I'm a PhD student at the University of Quebec in Montreal, and I also work at the River Institute. So this session will have 4 presentations, which should last 15 minutes, followed by a period of questions. If you have any questions, please use the Q&A box, and after the talk, I'll be able to read your questions. And during this event, if you have any technical issues, please email support@riverinstitute.ca. And just to note that this session will be recorded. So our first presenter for this session is Elizabeth Grater. She is from the University of Quebec at Trois-Rivières. And her presentation will be about bacterial metabolic journey through the St. Lawrence River riverscape. So Elizabeth, I'm going to pull your presentation. Share it with you.
Great.
There you go. So you should be able to have control now.
Yes, it looks good. Okay. Hello, everyone. Like I was introduced, my name is Elizabeth Grater. And I am currently finishing up my master's degree at the University of Quebec at Trois-Rivières. And I'm really happy to be here. That was a great presentation that we just heard. And I'm really excited to bring it back to the St. Lawrence on the other side of Canada and take you on a little metabolic journey through the St. Lawrence River. So large rivers are vast, important ecosystems that connect the land to the ocean and bring large amounts of terrestrial material to these systems that are then processed and go downstream into the oceans. They're also very important for sustaining human and natural ecosystems, but they're very complex. And so often it's hard to understand the whole functioning of a large river from the start to the end. And scientists often struggle to conceptualize the entire river or to really understand the functioning. And so what we mean by functioning is looking at the activity within the river and how that relates to the physical and chemical aspects of the ecosystem. So what I want to try to do and what I want to talk about today is, can we understand bacterial metabolism through the lens of, can we understand large rivers through the lens of bacterial metabolism? So can we understand the functioning through that lens? So why bacteria? Why are we focusing on bacteria today? Well, first, they are responsible for the largest flux of carbon on Earth, and they're very important reactors of materials. So all those materials come into rivers, they process them, they change them, and they alter the ecosystem around them. They're also very dynamic and sensitive to environmental changes. So they're a great tracer of both human impacts on ecosystems and natural impacts and changes. And they also are the base of a food web. So everything that builds off of river systems and goes up within a food web usually starts out with bacteria. And we can understand a lot by looking at bacteria. So to give you a little bit of background, I wanted to start out by looking at what happens when material enters into an aquatic system and how bacteria interact with those materials. So first, we start out with the input of dissolved organic materials. So what that is basically materials coming from the land or upstream, and those materials can be split into 2 different types. We have terrestrial materials, so you can think of sticks, you can think of leaves, you can think of things leached from soils, and those are usually more complex structures. And then we also have materials coming from phytoplankton, from algae, things like that, and they're smaller, simpler structures. And within that larger pool, we can pinpoint carbon as a really important aspect of the dissolved organic materials. And within that, associated to these materials, we also have nutrients that play a very important role. So next, I want to look at the carbon consumption. So this material enters into the ecosystem, and then bacteria consume that carbon, and they can apply it for multiple different aspects of their functioning. And what I want to focus on is the consumption of carbon and then the utilization for bacterial respiration or bacterial production. So bacterial respiration is when the bacteria consume the carbon, utilize it for energy production, and then respire out CO2. Or they can utilize that carbon for biomass production, so their growth, their cell production, and then those cells can actually go up into the food web. They can be food for zooplankton, then fish, and then eventually us. So we have these two different allocations that either can end up going as CO2 into the environment or go up the food chain as biomass. And it's important to understand that dynamic. So do bacteria use more carbon for respiration or production? And that can give us a key insight into the functioning of ecosystems. And one way we can look at that is by looking at bacterial growth efficiency. So how efficiently are bacteria utilizing carbon for that growth? And so this gets to my goals of my study. I want to assess the bacterial metabolic responses throughout the St. Lawrence River and identify possible drivers of this activity so we can get an idea of the functioning within the St. Lawrence River. So what I'm going to do is I'm going to look at the riverscape within the St. Lawrence through the lens of dissolved organic materials, bacterial metabolic responses, and then some possible drivers of those metabolic responses. And then the goals, the big questions are, what does bacterial metabolism tell us about the functioning of the St. Lawrence River? And what are possible factors driving these patterns? So briefly, I want to go through my methods that I performed for my master's. I was a part of two campaigns along the St. Lawrence River. There was one in 2017 and one in 2018. And I was able to participate on Mission Lamsalis aboard the Lamsalis research vessel. And in 2017, we targeted a smaller portion of the river. So if we look on this map, we focused on the area from Montreal to Trois-Rivières for 45 different sites. And then in 2018, we went all the way from Lake Ontario to Trois-Rivières for a total of 60 sites. And at each of these sites, we targeted a lateral transect of the river to really be able to capture the different water masses that are actually within the system. So the unique morphology of the river allows for the water to separate laterally, where we have the main channel, which makes up the bulk of the St. Lawrence River, originating from Lake Ontario. And then we have the northern channel of the river originating from the Ottawa River that stays pretty much in the northern section of the river and gets inputs from other tributaries. And then we actually have a mixed channel that starts right after Montreal, where they dump the sewage effluent of 3 million people there. And so this effluent gets transported downstream between these two main water channels, and the plume gradually gets diluted as we go downstream. But we want to understand the dynamic of that important hotspot as well. And one thing also I would like to mention before moving on is that we had two different hydrological conditions during these 2 years. So during 2018, we had relatively normal flows within the St. Lawrence River. The water levels were pretty much average to the historical flows. But then in 2017, we had historical highs. So very high water levels, very high flows throughout the St. Lawrence River. And that's something that I want to talk about in my research. And so our sampling campaign, we were able to use a GoFlow sampler to collect water from each of our sites. And then we did filtrations for our analyses. And for carbon and DOM, we used GFF filters, so very small pore sizes to take out anything living from the water samples. And then for bacterial metabolism, we used a bigger pore size to take out anything that could potentially consume the bacteria, so grazers and things like that we don't want in our samples. And so briefly, we looked at, like I said, the dissolved organic carbon and dissolved organic materials, and we took the concentrations to see how much carbon is in the system. We also wanted to know what types of dissolved organic material. So is it that terrestrial material? Is it that aquatically produced? So what type of materials? Using fluorescence and absorbance analysis. We also took concentrations, so how much phosphorus and nitrogen is in the system. And then we also looked at bacterial respiration. So how much are they breathing? How much oxygen are they consuming? And how much CO2 are they producing within closed ecosystem incubations? And then I also looked at bacterial production. So how much are they growing? How much are these bacteria growing? And we used a radioactive tracer of growth for that. So now, finally, I want to get to my results. So here we have the dissolved organic carbon, the DOC, within the St. Lawrence River. And this looks at the DOC concentrations from the Lake Ontario to Trois-Rivières. And here I have it split up in those 3 channels that I mentioned before, as well as the 2 different hydrological regions. So the straight lines with the circles, those are baseline flows, baseline water levels, whereas the triangles with the dashes are high flow conditions. And right away, if we look, we see that, okay, there are some peaks in concentration, but the concentration of DOC is relatively stable throughout the entire river, about 2.8 milligrams per liter as an average. The one thing we can highlight right here throughout the entire river is we see a peak in concentration at the Montreal effluent. So even though the Montreal effluent is a small amount of water, it is having a significant influence on these DOC concentrations. And it's also important to note that on average, we have significantly higher concentrations of DOC during those high flow conditions compared to baseline conditions. So next, we're looking at the type of dissolved organic matter. So we used optical properties to characterize the size and the characteristics of the dissolved organic matter, and we were able to pinpoint 5 different components or types of dissolved organic material that were in our system. And so I want to split those up into the different types. So we have 3 of these components, component 1, 2, 3, that's terrestrial derived, coming from the land, in more complex structures, where components 4 and 5 were more locally produced by algae and phytoplankton and were more simple structures. So if we plot those on a PCA, principal component analysis, along with the DOC concentrations and the concentrations of nutrients, we see really interesting trends start to come forward. So briefly, we have the main channel sites as the circles, the northern channel sites as the triangles, and then we have a gradient from upstream to downstream, going from lighter to darker. But right away, regardless of that, we see that there's a really distinct trend between the north and the main channel in both of the years, according to the composition of the dissolved organic materials and the nutrients. But if we focus here on baseline conditions, we see an interesting trend where they start out very separate, these 2 water masses. But as they go downstream, the composition of the dissolved organic material starts to converge, and they start to become more similar as we go downstream. But then if we look at high flow conditions, we see very distinct separation throughout the entire river with none of that convergence occurring. So this suggests that the materials are just flowing downstream and there's little mixing occurring compared to the baseline conditions. So finally, I want to get to the bacterial metabolism that I had promised earlier. So here we have the carbon consumption as our top figure, and then we have the growth efficiency, which the growth efficiency relates to the biomass produced. And again, we have the different water masses and the different hydrological years. So if we focus on the bulk of the river, so that main channel, we see that there's an increase in both carbon consumption and bacterial growth downstream. We also see relatively similar rates of consumption and growth efficiency within the main channel and the north channel, but we've noticed that the Montreal effluent here in the mixed channel has extremely high rates of bacterial activity, actually 5 times the rate compared to the rest of the river. And so right when that effluent enters the river, there's a dramatic increase in bacterial metabolism, but then downstream we see that it goes back down to those levels that we're seeing throughout the river. And then also one thing to pinpoint is that during high flow conditions, we actually saw less bacterial metabolism compared to normal flow conditions. So if we remember back, for the DOC concentrations, the carbon concentrations, there were higher levels of carbon during the high flow conditions, but now we're seeing lower bacterial metabolism during high flow. So this means that more of that carbon is being transported to the estuaries unprocessed without reacting to bacteria. And so next, I want to look at the drivers, the possible drivers of bacterial metabolism. So what we did here is we plotted the 4 facets of bacterial metabolism with those the composition of organic materials and nutrients, and we found a really interesting trend. So if we focus on A here, we see that bacterial metabolism had a very strong relationship to the terrestrial materials and nutrients coming from the system. And we found a negative correlation with the other types of carbon, the carbon produced locally. And what we did is we took the scores along PSL1 and we plotted them, so bacteria versus the carbon from that initial axis here, and we plotted it. We found a beautiful linear relationship between bacteria and carbon consumption. So we found very strong correlations between those inputs of material and the changes, the terrestrial material, with the bacterial metabolism. And then if we go ahead and look at high flow conditions, we see similar trends, but to a weaker degree. So we see that this correlation between bacteria and the inputs of material is weaker during high flow conditions. And so in conclusion, I want to wrap up and just say that we found some interesting trends within the St. Lawrence River. And this is just the baseline, the first steps of understanding the functioning of this large river. And so what we found was that throughout the river, we found pretty stable concentrations of DOC, yet we found dynamics in the DOM composition and the bacterial activity, suggesting that there's some decoupling occurring between those two reactors of the St. Lawrence River. We also found an increase in terrestrial DOM downstream and an increase in bacterial activity downstream, which is actually the reverse trend of what is normally found in large rivers. And we think this is due to the really unique headwater system, that large Great Lake within the St. Lawrence River, creating this interesting new type of ecosystem compared to other large rivers. We also found that hydrology played a very important role in controlling both bacterial activity and DOM loading, and that Montreal and other areas like fluvial lakes were important hotspots of activity. And so we found that bacteria was a really good tracer of the different hotspots of activity, as well as the changes in hydrology that we were seeing. And what I wanted to end with was just highlighting some other research that we are conducting on the St. Lawrence River. So it's now been 4 years that we have been doing research on the St. Lawrence, and we've looked at a variety of things, such as pharmaceuticals, E. coli, DNA sequencing, isotopes, heavy metals. And this year we were able to go all the way to Rimouski to look at the estuary, and we hope to gather this data together and do a good assessment of the St. Lawrence. And with that, I would like to thank everyone involved in my project, acknowledge the lab that I'm a part of and my co-supervisor lab, and thank all of the wonderful research group that I've been a part of, all the wonderful cruises and the crew as well. And with that, thank you for listening and participating to my presentation.
Thank you, Elizabeth. We have time for maybe 1 or 2 questions. I can start with my question. I'm just wondering, like, if we put this in a climate change scenario, do you think that some other factors, like, I don't know, water temperature could become more important than hydrological factors on bacterial activity?
Yes, I mean, we've found that, we haven't personally found, but in my research, I've found that temperature does play an important factor. And right now, the water temperature of the St. Lawrence, from what we've been looking at, is pretty stable. We haven't seen a whole lot of fluctuations, but if that water temperature did increase, then we would expect to see increased bacterial metabolism. One thing that I wanted to focus in terms of climate change is that we're actually expecting more flooding events. And these changes in hydrological events occurring, like we saw a big flooding in 2017, and since then, these big flooding events. And so we can see now that there's less bacterial activity occurring during high flood events. And so we're expecting to see higher loads of materials entering the estuary, which could potentially have some negative effects and create some acidification or hypoxia within these systems. So we see the importance of both, yes, hydrology, and it is a good point, water temperature being an important factor. Yeah.
Now there's a question from Marianne Racine. She's asking, did you also happen to look at the differences in communities during those sampling events?
Yes, so I looked both at abundance and I also did DNA sequencing, and we did see some differences in the community structures throughout the river. I also looked at the metabolic capacities, and we found that within the different water masses, there were different communities, which is expected because we have different DOM and organic materials coming forward. And we saw very distinct trends coming from Montreal. So we're trying to target that sewage effluent because we want to see what kind of unique bacterial communities and viruses and pathogens are entering the system from that sewage treatment. But we want to do more sequencing in the future to get a more pinpointed idea of those communities. Great question. Thank you.
All right, thank you so much. And it's a great presentation. And I feel like the microbial loop is always like underlooked, but so important. So thanks.
Good. I agree. Thanks a lot.